there any way I can check and see if it's properly directed at you?
That's what I'm discovering right now.
Okay. That's good enough. Pretty good.
That covers the full.
Yeah. Sweet. Good recording already?
Yeah.
Crayons, sir.
Report for like 2 hours, so pretty good.
Good, because your talk is only going to be about an hour and 20 minutes, so it's good.
Alright, today we're going to be talking about the CATH database, and probably wondering what the CATH database is. Well, a database is usually a collection of something, and the CATH database is a collection of protein domains, and that's actually what we're just looking at, different domains of proteins. And why use CATH? You can use it for research. If you're dealing with a protein, you can run the domains and find out different information. You can use it for medical because drugs deal directly with proteins and enzymes. Evolution, comparing proteins that are in humans and chimpanzees and their different functions, from functioning protein in a fish to functioning protein in a human. Look at the different motifs of the proteins that way, functions. And I'm going to show a movie trailer actually, because what if there was an epidemic? What would scientists do if there was an epidemic?
Let's watch the Twilight trailer instead.
Yeah.
Dynamite film.
Yeah, the volume should be right on the front of those.
I wanted to mention I saw this trailer and I saw him playing with the high mall thing on it.
I think that's
what
why
I was mentioning.
Yeah, indeed.
Oh, it's still your thunder.
Volume's all the way down. Crank it. There we go.
Cleaning ceremony for manufacturing.
She mentioned seeing anyone who was sick? Anyone on a plane to the airport?
No, she said she was jet lagged.
The average person touches their face 3 to 5 times every waking minute. In between, we're touching doorknobs, water fountains, and each other.
Go up to your room, honey. So we have learned no treatment and no sleep this time.
You had a seizure this morning, Beth.
She got a history of seizures and allergies.
As of last night, there were 32 cases.
Unfortunately, she did die.
Lisa, can I go talk to her?
Mr. Olof, your wife is dead.
What are you talking about? What happened to her? What happened to her?
Is there any way someone can weaponize the bird flu as our delivery? Someone doesn't have to weaponize the bird flu. The birds are doing it.
Watch this. It's transmission, so we just need to know which direction.
On that one, there were 2 people, and then 4, and then 16. In 3 months, it's 1 billion. That's where we're headed.
They're calling out the National Guard. They're moving the president underground. People will panic. People tip over.
The truth is being kept from the world.
Cook your samples, destroy everything.
I never saw so many big stars in one movie.
Yeah,
You can't
yeah,
have
that's
all
true.
those. Jude Law, Lawrence Fishburne,
Services room.
Winslet, Gwyneth Paltrow.
I think this came out September 9th, and I've been meaning to see it, but no
Oh
time.
yeah, I need to see this movie.
Don't talk to anyone. Don't touch anyone. Stay away from
That guy
her.
was in the East Woods.
It's figuring us out faster than we're figuring it out. It's mutating.
And so certainly one would have to wonder what would take place was an epidemic? Well, one of the ways is if they could actually obtain the virus and find its code for proteins or bacteria's code for proteins, they could use CATH database. It has 35 protein structures and or not 35,000 protein structures and over 100,000 domain chains in the database. How CATH algorithms work. First, if you were to select a sequence of amino acids.
If you don't want to block the screen, you can take it all the way to that side, or if you can stand blocking the screen for a few minutes, you can put it right there.
I can block it first.
There. And then when you're done, maybe take it to that left over that side. Then that will still be available to you when you need it.
Yeah.
That's the best thing I've found.
Everyone? Okay.
You can swing left side toward us a little bit. That'll help us on this side. Good. That'll do.
So we have CATH. And CATH is basically made of thousands and thousands and thousands of protein domains. And what you need to do is you have a amino acid sequence, and you need to go into this database using an algorithm that CATH has. And
So this would be some amino acid sequence that we don't know what it does.
yes,
Like for
correct.
example, there's an epidemic. We've discovered, we've sequenced some genes.
Correct.
We don't know what these amino acid sequence code for, so we plug them in
And
here.
you've obtained the various proteins, and you're going to sequence it against the database because there can be various things that can help you out there. The first node in a tree, it's going to branch out as a tree, is called class. And there's actually 4 routes you can start out in. And as Dr. Freeman was talking about on Tuesday, there's alpha helixes, there's beta strands or beta sheets. Or if it has a series of bolts, it can fall into that. Or if it has motifs, it's going to fall into that category. Like I said, the class is the overall secondary structure of the domain. The second node is architecture. And as displayed on the PowerPoint, architecture, it can be like a tin barrel, or a sandwich, or a roll. And obviously, as you can see, it selected the alpha and both beta, and it's derived from there.
Okay, so there are architectures that are typical of all alpha. A protein that's almost all alpha might have various architectures it could fall into.
Correct.
A protein that's almost all beta could have a bunch of architectures, and then some that are alpha and beta will also have. This is suggesting, and this turns out to be true, it's suggesting that there aren't an unlimited number of protein structures in the world, that they fall into particular categories.
Orders and classes, correct.
Sort of taking advantage of that evolutionary situation.
An important note is architecture is high structural similarity, but no evidence of homology. That takes into consideration species if it's homology based. This solely looks at structure and functionality. It doesn't care if it comes from chimpanzees or fish. If the protein has a similar structure, it's going to fall into that node of the tree. The third node is topology. For the second, you have architecture. This is a more refined. It's going to find a certain topology and go there. It's as you can see here. The sandwich, it can be in various forms. It can be inflatodoxin or beta-lactamase.
So these are names. Those are names of particular proteins that are typical examples of this architecture.
Sure.
So is that right?
Yes.
So I'm learning this,
Yeah.
but because I have a thicker amount of information to plug onto it, I'm making comments so you guys can. I'm asking these questions. I'm asking are questions. They're not like trying to add stuff. I'm guessing that flavodoxin is a particular protein, and if that particular protein has a lot of topologies that are like it, it will recognize it's the flavodoxin type, or the beta-lactamase type of architecture, or sorry, type of topology.
Yes, these two will actually have domains. They're just examples of what the topology would be.
Okay.
Yeah, I think that's the best way to say that. A fourth node goes back to homology. It's going to find, this is where it takes into place some consideration of species. If it goes through the first three nodes and it's also similar to like a chimpanzee, it's going to find a similarity between that, and the algorithm's going to go that direction. After that, the 5th, 6th, 7th, and 8th and 9th nodes, they're all sequence related. That means the algorithm's going to run, it's not going to really take any consideration unless if there's a certain sequence of amino acids, it's going to narrow it down. And when we go to the website, you'll actually see how it does. But it goes down to 5 different levels, and then it'll actually find the best suited domain.
Do you know, are you going to tell us what those letters mean? So
I've tried to find it, it's all just sequencing. And I've tried to find what those letters are, but it doesn't say. It probably has to do something with the people that wrote the algorithm, I'm guessing. But it's all sequencing of matching different sequences. If there's like a poly A section of the DNA and that codes for something, it's all sequencing. That doesn't actually mean. I'm going to have you guys go to NCBI. And it's not ncbi.com or .org, it's, you just go to Google, type in NCBI.
this way.
Okay. Instead of in search of all databases, we're going to click on proteins and type in pretty much any protein that you like.
Okay, wait again, just make sure everybody's with you here. Did everybody switch all databases to just protein? So searching just protein, and you want us to type insulin?
That's what I'm going with. You can type in whatever protein you like. I'm going to go ahead and you can select any of these. They're different species. I'm going to go ahead and pick 13. This one's good. And if you scroll to the bottom, this is the amino acid sequence. I'm going to use this, and I'm going to actually go to the CATH website. And go ahead and in Google type in CATH database, it'll be probably the first thing that's there.
So you can either scroll down to the bottom of that page and copy the amino acid sequence, or you can click. There's a little button called Fast A, and that will give you nothing but the amino acid sequence. So we're copying that, right?
Yes, you can copy that. You can copy the numbers and everything. You don't need to worry about that. Now, I'm taking this amino acid sequence off of NCBI, but if you're doing research and you were inserting a gene into a bacteria and then extracting that and seeing if you have the correct protein, you could also see if you have the right code you can run it against the sequence. And if you get a different, you might get a variant, which will lead to a mutation in the protein. And then if you have a mutation in the protein, you'll select your sequence, but for now we're just getting the insulin sequence just so I can show you. But it will actually